Hi everyone, and welcome to today's edition of Biology 206. I'm Dr. Jennifer Baltzer, and today we're going to be learning about coping with environmental variation. So in the last couple of lectures, we've been looking at environmental gradients at sort of the planetary scale, at the global scale, the implication of those environmental gradients for the distribution of biomes, which is, largely characterized by the dominant vegetation types. And today we're going to be learning about how those mechanisms by which those individuals within populations, within species, are able to cope with different components of environmental variation related to temperature and water. So precipitation and temperature, which we've been talking about a lot over the last couple of lectures. Okay, so what will we learn in this lecture? We're going to be able to better understand why the physical environment is the ultimate determinant of species distributions. What are some of the different ways that organisms regulate temperature and water potential and the regulation of water balance in plants and animals. So these are, the two key environmental gradients for survival: temperature and precipitation, or water availability, and they interact in lots of ways as well. So we're going to be learning about those things today. Okay, so when we think about where organisms occur, a lot of times we need to think about how do they deal with what would be considered environmental extremes for them. So, if we think about the boreal biome, which we learned about last week, it has some really extreme temperatures, right? So it goes from -30 in the winter to +20 in the summer, or even warmer. That's a 50-degree Celsius range variability in temperature. And you have some organisms that can't move in response to those changing temperatures, and so they have to be able to deal with temperature in situ, changes in precipitation in situ, etc. So there's a whole range of environmental extremes or environmental stressors that organisms are faced with, and this depends on the organism. What is extreme for one organism is really just, kind of middle of the pack for another organism, and so it's really has to do with the adaptations of those organisms, and that's what dictates their ability to deal with these different environmental conditions. So when we are thinking about some of these environmental variation and response of different taxa to environmental variation, we talk about different life history strategies and the ability of different species to deal with environmental stress. So there's a couple of key strategies. These are not all of the strategies, but when we think about stress, you can either avoid it. So a response to avoidance is a response to stressful environmental conditions that lessen their effects through some behavior or physiological activity that minimizes an organism's exposure to the stress. So you modify how you're behaving or how your physiology works in order to lessen the impact of that stress. Tolerance, on the other hand, is the ability to survive those environmental conditions. Okay, so we'll get into some examples of these, but there tends to be a spectrum from this sort of avoidance, or should I stay or should I go, I guess would be a good analogy, but this ability to modify how that stress is acting on you. And so a great example, if we think about desert environments, you have some plants that are able to, they have physiology and more physiological mechanisms, morphological structures that allow them to tolerate the low water and high temperature conditions of the desert. So an example of one of those adaptations is, physiological, photosynthetic pathway that allows them to open their stomates and fix carbon at night, and then store it away until the daytime when the sunlight is shining on the photosynthetic tissues. And at that point, they actually fix that carbon, convert that carbon dioxide into sugar. And so this, opening their stomates when it's coolest and most humid at night to bring in the carbon dioxide so they minimize water loss, and then lock that carbon away until they're able to actually fix it into sugars. Okay, in contrast, avoidance in the same desert environment can look like plants that complete their entire life cycle in the little window of time in which you have precipitation. And so they will go from seed to seed when moisture is available in the desert, and then become dormant during those times between during the dry period that dominates much of the year in the desert. So those are just two examples of avoidance versus tolerance. But it's really important to think about these life history strategies when we're contemplating how organisms respond to. Okay, so moving on to environmental tolerance. This is very similar to a graph that is in your text. This is taken from a different text and just shows a little bit more information. But what we are looking at is gradients of low resource availability to gradients of high resource availability. This could be water, light, nutrients, what have you. And on the y-axis, it's low population abundance to high population abundance. And for any given resource gradient, you'll see different species responses to that gradient, and even differences amongst populations where you've had ecotypic divergence. And so what happens is you have a zone where the resource conditions are really optimal for different physiological functions. So this is the area where growth, reproduction, all of these kinds of measures of fitness are optimized, and your population will, as a consequence, be high. You have the greatest abundance here. Now you move into these zones of physiological stress. This is where different physiological processes are not functioning in an optimal way, and so those same fitness-related measures are reduced. They're dampened, and we see this situation where we have lower population sizes in response to areas of low or high values of that particular gradient for that species. And then you move into the zone of intolerance. This is where the species is not present. So this is outside of the set of environmental conditions that species can actually occur in. Yeah, and so when we think about resource availability gradients, this is the kind of response we expect. Some ideal conditions, some conditions that are physiologically stressful, and then a set of conditions where the species cannot tolerate the environmental extremes. Okay, so next we move on to the concept of the niche. This is a really important ecological concept, and so when we think about those environmental gradients we just looked at, we can think about them in the context of, where a species is able to occur and where its distributions tail off. So those different zones that we looked at in the previous slide. But we can think about this in the context of biotic interactions too. And so when we think about the fundamental niche, this is the full range of conditions and resources that allows a species population to survive and reproduce in the absence of competitors and predators. So in the absence of those negative ecological interactions. And so this is just its response to the entire environmental gradient. And this blue line represents that potential distribution or the fundamental niche, right? And this is really simplified because this is talking about a single environmental gradient, but when we take all of the environmental gradients in the environment and we map them together, this is when we actually get the niche. But to simplify it, we'll think about it in this sort of two-dimensional space, the abundance of an organism as a function of the environmental gradient. Then the realized niche is the full range of conditions and resources that allows a species population to survive and reproduce in the presence of competitors and predators. So when we take into account those negative ecological interactions, all of a sudden that zone is reduced, because if we go back to this, remember there's this zone of physiological stress. If you add a layer of biotic stress onto that, so maybe you have predation, maybe you have competition, all of a sudden individuals may not be able to occur in those areas. And so all of a sudden your actual realized distribution is smaller across this environmental gradient, and overall your abundance is lower across all portions of that gradient. Okay, so fundamental niche versus realized niche. The realized niche is always smaller, either the same size or smaller than the fundamental niche. You never have a realized niche that's larger than the fundamental niche. Okay. And this is a great example of this. This is sort of a classic example where we can look at the distribution of these different barnacles in intertidal zones. And so we have two species of barnacle, Thalamus and Semibalanus. And Semibalanus is the large ones down here below the low tide mark. And Thalamus is these smaller ones, the smaller yellowish ones, above the low tide mark. Okay, and so we have these two species co-occurring. Those two species are using similar resources, and so what they would be doing if they're co-occurring, they're sharing resources, right? So they're competing for resources. And so what we see happening is when Semibalanus is not present, Thalamus occupies the entire zone from high tide all the way down to the low tide mark. Okay, so we see its distribution is greatly expanded when Semibalanus is absent. When Semibalanus is present, its distribution is restricted between the low tide and the high tide environment. And so we can see that this is its, you know, assuming that there are no other negative biologic or negative biotic interactions, we can think of this as its fundamental niche. Whereas when it's in the presence of this competitor, Semibalanus, its realized niche is much, much smaller than its fundamental niche. Okay, so this is when we talk about this reduction in its distribution, has to do with those processes. Okay. Yeah. And then we can think about this in the context of entire species distribution. So in this previous example, this is sort of a local distribution, right? We've got differences in the local distribution of this species as a function of the presence or absence of this species. Now we can move up to entire ranges as well and think about where, you know, the different factors that contribute to the distribution of a species across these, you know, temperature and precipitation gradients. And so again, we can think about that in the context of, well, what are the conditions across which, in this case, aspen is able to occur? So that would be the predicted distribution, which we see here in green. And then we can also look at, well, what is the actual distribution? Where do we actually find it? So if you run, you know, again, if you run experiments of what conditions species are able to occur across, or in the case of a widespread species like aspen, what are the full range of conditions that it exists in across its entire range? And that would lead us to expect that it could occupy a broader range of conditions than it actually does. Its actual distribution, you can see, is smaller than its predicted distribution based on those climatic factors. And so when we think about species distributions, you often hear the term climate envelopes, and these are the range of climate conditions under which a species occurs, or its climate niche. Okay, and again, that can be constrained by biotic interactions that alter the range of that particular species. Okay. And when we think about climate change forecasts, climate change projections, and their impacts on the distribution of species, it's also important to realize that even though the temperature and precipitation may be changing in a certain way that could favor or reduce the ability of that species to be in a particular location, if the biotic conditions that it's experiencing are also changing, this can modify whether or not we'll see that expected response. So a great example of that is up at tree line. There's some really nice growing evidence that even though, even though the climate has warmed sufficiently that Arctic tree line could have advanced in many places, there are lots of locations where this simply is not happening. And experimental studies have shown this to be caused by seed predation by small mammals that live in the tundra environment. So you have seeds of trees being arriving in tundra environments, but those seeds are delicious snacks for the rodents that occur in those environments and can, as a consequence, that biotic interaction, that granivory, is limiting the expansion limiting to some extent the expansion of those trees in tundra environments. So this is, biotic conditions can really constrain how climatic factors can, how well climatic factors can predict where a species is going to occur. Okay. So we've already talked a little bit about stress, but I just wanted to define it here. Stress is the condition in which an environmental change results in a decrease in the rate of an important physiological process, thereby lowering the potential for an organism's survival, growth, or reproduction. Okay, so when we have a stress, there are a couple of ways that when we have a consistent stress, so a short-term stress may not modify, you may have some, actually, let's just go to the next slide. You're going to have, when you have some stress imposed, so you've got, here we have rate of physiological process from low to high. It's been bopping along like this, responding to sort of short-term changes in its conditions. But then a stress is imposed. And maybe this is drought, maybe this is a long-term drought, maybe this is, an introduction of a species. Maybe, there's any number of examples of where a stress may come into play. Maybe it's a pathogen. But a stress is imposed on the system, and all of a sudden that goes from, that particular species goes from functioning in an optimum way to crashing down here to some low rate of physiological process. And this happens over the course of, over a short time span, the course of minutes, days, weeks, depending on the process you're talking about. Okay. But as you move through, and if that stress is continually imposed on that organism, it is going to start to do what we refer to as acclimation, or the adjustment of physiology, morphology, or behavior to lessen the effect of an environmental change and minimize the associated stress. So our bodies respond to stress, and we do things that change that stress level, and our physiological responses can be modified to minimize that stress as well. So this is acclimation, or we talk about plastic responses. These acclimatory responses occur at the individual level. And so these are behavioral or physiological responses at that level, so within an individual. But if a population is exposed, if an entire population is exposed to that same stress over some very long period of time, we will see what we refer to as natural selection. So those individuals that have behavioral, physiological, or morphological responses that really help to reduce the stress and improve the outcome for that individual, individuals that have those abilities will be selected for in the population. And over the course of generations, then we may start to see a change in the way that population responds to this particular stress. And this is what we refer to over the course of generations and evolutionary time. This is what we refer to as adaptation, a physiological, morphological, or behavioral trait with an underlying genetic basis that enhances survival and reproduction in that environment. So through the course of generation, those individuals that are able to that have the ability to respond to this stress and improve their situation will be selected for in the population, or that have traits that give them some advantage in that new stressful environment will be selected for, which through long periods of time leads to this adaptation. So this is an evolutionary process. This is an individual plastic process, and it's really important not to mix these two terms up. Sometimes people tend to use them interchangeably. That is not okay to do in the context of ecology. When we talk about acclimation, we're really very specifically talking about those individual level processes. When we're talking about adaptation, it's that longer term process of selection by of adaptation by natural selection. So natural selection acting to favor those individuals that are better suited to deal with that stress. Okay, so two key stressors that organisms have to face are temperature and precipitation. And so we're going to go through temperature and then we'll shift to water availability. But when we think about temperature, we can think about all of these different conditions that organisms have to face. So everything from, very cold temperatures, where you have, sub-zero temperatures, and you have, freezing temperatures that organisms have to be able to cope with, and we'll talk about how some organisms do that in this lecture, to very sort of temperate conditions that are within the reasonable ranges of body temperatures for certain organisms, up to kind of fairly warm temperatures that match the body temperature of active birds and mammals, up to incredibly hot temperatures when we're thinking about, for example, hot springs, bacteria and archaea, or organisms that are able to live in these deep sea thermal vents, for example. So really, really hot, harsh environments. Okay, so we run the whole gamut of temperatures that have led to all kinds of different amazing adaptations to deal with temperature, and we'll talk about some of those today. But when we think about temperature, we can, we know, just like any environmental gradient, so this is exactly what we saw in those physiological tolerance curves. So we have leaf temperature going from 20 degrees up to 50 degrees. We have three different species depicted by these three colors. These are three tropical species: Ficus, Acroma, and Calophyllum. And we can see their physiological response curves. This is photosynthesis in micromoles of carbon per meter squared per second. And this is respiration in micromoles of carbon per meter squared per second. And what we see is that photosynthetic rates are lower at lower temperatures. They increase to some optimum, and that's pointed out by these big fat arrows. And then they continue to decrease as temperatures increase. And so you start to have, the enzymes that govern photosynthetic processes start to not function as well when temperatures are too high, for example. And these optimal temperatures differ between species, and that's not surprising, and it's a really important component of why different species are in different environments. These are all admittedly tropical species, so you can see that for most of them, their optimal temperatures in around 30 degrees C leaf temperature, which is pretty on par with temperatures in the tropics. Respiration continues to increase continuously as we increase leaf temperature, and so you see this situation where you've got leaf temperatures increasing, and both photosynthetic and respiration rates increasing. This is what we would expect. But then photosynthetic rates start to decline, and respiration rates continue to increase. Okay, so we have this problem that if you get too hot or too cold, you don't function very well. And there's all kinds of good physiological reasons for that. And if you're interested in those, there's all sorts of amazing physiological courses you can take for both animals and plants. But we are going to very briefly get into how do organisms regulate temperature, because we want to avoid these really high and really low temperatures Okay, so when we think about the energy balance of an organism, there's a number of pieces we need to think about. And this is delta H, so change in heat is a function of shortwave radiation. And so that's the actual radiation, the direct radiation from the sun. The infrared radiation incoming and the infrared radiation outgoing. And so this is infrared is long wave radiation, so it's heat being emitted from some other source. Let's think about standing on the pavement in the summer, and you can feel the heat kind of being radiated from that black surface. Our bodies emit heat all the time as well. So you've got long wave radiation coming in, long wave radiation going out. Okay, so next we have convection, which is heat carried away by cool water or air moving across a warm body. Okay? And so you can think about this when, for example, when you're hot and you jump into a swimming pool, or you're hot and you're in a place that has, that you don't have windy conditions, and then you step out into an opening and all of a sudden a nice breeze passes by you. So this is our convective heat loss. Conduction is our next step, and this is direct transfer of energy from warmer, rapidly moving molecules to cooler, slowing moving molecules. And then finally, we have, in the context of plants, and this is also one for animals, but evaporative cooling, so latent heat transfer resulting from the change in state of water from liquid to vapor. And so when we're thinking about plants, this is transpiration, so stomates opening and water evaporating from those leaf surfaces, cooling the leaf as the water is carried away. When we think about us as humans, this may be water evaporating from our bodies as we sweat, heat loss from, again, water evaporating from our mouth as we breathe. These are all ways that we have evaporative heat loss from our bodies. And you can think about that, the mouth one, for example, with dogs when they pant. This is a key way that dogs lose heat to evaporative cooling. And then finally, there's metabolic heat generation in animals. And we'll get into this when we talk about endotherms and ectotherms. But when we have endothermic animals that actually generate heat to maintain their body temperature, so they use metabolic activity to generate heat and maintain their body temperature at within a fixed range of temperatures. And so we're an example of that. Our body temperatures are typically right in around 37 degrees Celsius. That's a very costly thing to do, to have a body that can maintain temperature. And so this is a really important piece of, endotherms being able to occupy all kinds of different environments on the planet. Okay, so these are the different pieces when we think about this heat balance and different places where we can manipulate temperature to better, to reduce stress from the surrounding environment. Okay, so transpiration is, as we just talked about in plants, a really important one. This is an electron micrograph of a stomate. So stomates, if you look at a leaf surface, they're covered in these pores, which are called stomates, and these pores are controlled by a variety of things in the plant, including various hormone signals, and the guard cells cause the stomates to close and to open. Okay, and so when a plant is taking up carbon dioxide, the stomate is open, carbon dioxide is entering the leaf, water is leaving the leaf. This can be a problem for plants because too much water loss can be problematic when conditions are dry. This can also be a benefit to the plant because if the leaves are hot and you open your stomates, this can help you to cool your leaf surface because you have evaporative cooling happening on the leaf surface as a consequence of transpiration. And so we know from our previous 2 lectures ago as well that this evapotranspirative water flux from plant communities can be a really important cooling effect for the ecosystem as a whole. And you can see this impact on this infrared diagram, or this infrared image here. So this is an Arabidopsis plant, so Arabidopsis thaliana, a model plant organism, and Arabidopsis with its stomates closed. And here is our temperature gradient here. So when the stomates are closed, the temperature of the leaves is up here around, I don't know, 22.5 degrees Celsius, maybe closer to 23 or 24 at the leaf margins. When the stomates are open, we see that this temperature drops quite substantially down to, you know, somewhere in here around 21 or so degrees. And so we have this clear cooling effect of opening the stomates. When the stomates are closed, not as much heat is carried away. When the stomates are open, a lot more heat is lost due to transpirative cooling, or evaporative cooling, I should say. Now, how can plants modify incident solar radiation? So if we think about that energy balance equation, you have your incoming solar radiation. One way that plants can modify that, you sort of think, well, you know, plants are not like animals. Animals, if it's too sunny, what do? We get up and we move to someplace that's shadier. So animals can just move themselves into a shadier location to reduce incident radiation, so the direct solar radiation. How can plants modify this? And the answer is by modifying the reflectivity of their surface. Okay, and so one way that plants can do this is by having what we refer to as a pubescent leaf surface, or a fuzzy leaf surface. And so you end up with these fuzzy white hairs on the surface of the leaf. And if you examine some leaves, maybe you even have a host plant that has fuzzy leaves. That's one of the mechanisms that plants use fuzzy leaves for, is to help to reflect light from the surface. And so here we have 3 different species, 2 of which do not have this fuzziness, this pubescence. One, this middle species that does have pubescent leaves. And what we see is the percent solar absorptance, which goes from 0 to 90% here, is much higher in those 2 species that do not have pubescent leaves, and much lower in this species that does. And so by having this fuzziness, these little white hairs on the surface of the leaf, the plant increases its reflectivity, increases the albedo of its leaves, if you will. And we can see the same thing for summer versus winter leaves in this particular species down here. And so these are micrographs. And so here we have a seasonal response of pubescence, where our summer leaves have a much thicker hairy surface than our winter leaves, and this has a direct impact on the amount of solar radiation that's reflected. So here, instead of solar absorptance, we have solar reflectance. So a higher reflectance, we would expect a higher reflectance with the more pubescent surface, and that's exactly what we see. So this black line is the summer leaf, the green line is the winter leaf. The summer leaf has greater pubescence, and the infrared radiation and the solar radiation reflected from that surface are higher as a consequence of those little white hairs on the surface of the leaf. So even though plants cannot get up and move to a shady location, they can in some ways create some shade themselves. There are other things that this fuzziness or pubescence can modify. And so if we take a look at this snow lotus, the mechanism for this pubescence is actually to insulate the plant. So this is a snow lotus. It grows very early in the season, and so the temperatures tend to be fairly cold. And what happens is that by having this greater hairiness, instead of having a nice smooth leaf surface that air moves across really readily, that fuzziness helps to decrease the convective heat loss. And so it increases the boundary layer. It increases this boundary layer. So you have air that's flowing across here. The rougher your surface on the leaf or the plant, the thicker the boundary layer, and the slower the air is that moves across the surface. So that reduces convective heat loss and keeps this plant just a little bit warmer. And if we think about animals, fur thickness can play a similar insulating role. It's same kind of idea. You reduce that convective heat loss by having that added layer of fur. So when you have short fur versus long fur, the insulation value increases the longer the fur is. And so it's the same kind of idea that there are some additional complexities to it, but it reduces the ability for your body to actually transfer energy with lose heat to convective heat loss. Okay, so we've talked about a few things that plants can do to keep themselves cool, to reduce or to keep themselves warm in one case. But what other things do animals do? And animals, of course, have lots of other tools at their disposal. So one of the key things is this actual regulation of body temperatures. So endotherms rely primarily on internal heat generation, so metabolic heat production, that final term that was in parentheses in our equation a few slides back. An example of this, well, us, of course, but here we have our little bobcat. You can see that the bobcat temperature, regardless of the ambient air temperature, is maintained right up here around 37, 38, 39 degrees Celsius. That's incredibly consistent, and our bodies are the same way. Our bodies cannot tolerate variation in temperature, and we regulate our temperature internally and maintain it at a similar temperature as the bobcat. In contrast, here we have a snake, an ectotherm, which is admittedly consuming an endotherm. But the body temperature of the snake responds pretty directly. It's pretty close to a one-to-one relationship with the air temperature. Okay, and there are some ways that ectotherms can help to regulate their body temperature, predominantly through muscle contractions and movement of, primarily through muscle contraction. But for the most part, we have ectotherms that respond to external temperatures and so have to use other modes of staying warm. So these are our cold-blooded species. They regulate body temperature primarily through energy exchange with the external environment. So much more similar to those processes that plants have to face. Okay, so but unlike plants that are stationary and have sort of limited ways of modifying their temperature, their thermal conditions, animals have some extra tricks for modifying energy balance, and one of these is muscle contraction. So they gave a couple of examples in your text, and these are pretty neat examples. One is the example of these honeybees. When attacked by this giant wasp here, the honeybees will actually form a ball around the wasp and contract their muscles, and their contractions of their muscles generate so much heat that it can actually kill the wasp. It increases the body temperature of the wasp to a temperature that's unsustainable, and it kills the wasp. And so this just demonstrates the effectiveness of muscle contraction in heat generation. Okay, another example of this is in this tuna. And we have heat generation in swimming muscles, and then there's this kind of heat exchanger that's happening where you've got your venous blood and your cooler arterial blood. So your venous blood coming from the muscles, so it's deoxygenated blood that's coming from those muscles that have been contracting, and it's warmed up the blood, and it passes by the arterial blood and warms up that arterial blood. And so you have this kind of heat exchanger the heat is all generated by muscle contraction in the fish. Okay, and so you have these, gradients in temperature from these locations that are close to the swimming muscles to the outside of the body. And so really important mechanisms for heat generation and heat exchange in some endotherms. Second, of course, is behavioral changes. Animals can move, and they can move into sunny locations when they're too cool and into shady locations when they're too hot. And a really important behavior that a lot of endotherms display is this behavior of basking. And I also like to bask, and I sometimes think of myself as being cold-blooded. I don't stay warm very well, and so we all know how nice it is to kind of, when you're cool, to sit out in the sunshine. Lots of animals use that as a main way to warm up their bodies when their bodies start to cool down too much. And so this is really, really important for a whole range of ectotherms, including some in in aquatic environments. So I listened to a really neat podcast the other day where they were talking about basking sharks, juvenile sharks that actually come close to the shoreline to bask in the warm water. And so, and they were talking about actually documenting the suntans that these sharks were getting. But, you know, just to say, it's not limited to these kinds of organisms that we think of as basking organisms. There's lots of organisms that take advantage of moving into warmer areas, warm sunny areas, to absorb additional heat into their bodies. And then thermoregulation, which is metabolic heat generation. This is by far and away the most amazing trick. And so for any, you know, for endothermic animals, we have conditions where, you know, where you're in kind of the thermoneutral zone, where the temperatures are, the external temperature is within a reasonable range for the body to just kind of maintain its homeostasis, its constant temperature. When you reach some lower critical zone, all of a sudden the body actually has to generate additional heat to maintain that body temperature. And so what we see happening is once you cross that lower critical zone, that the basal metabolic rate begins to increase. So the metabolic costs associated with temperature regulation begin to increase because the body has to use a lot more energy to generate that heat that keeps the body at whatever temperature it is. And so we can think about this for different organisms. And when we're in this thermoneutral zone, sort of small behavioral modifications can help to maintain that body at its appropriate temperature. So, for example, moving into a warmer area, moving into the sunshine, moving from the sunshine into the shade. All of these things are sort of small behavioral modifications, maybe some small behavioral modifications that are fine for maintaining optimal body temperature. And then we move into past this lower critical zone, and all of a sudden heat loss is greater than metabolic heat production, and the body has to really increase its heat production to maintain body temperature. Okay, so if we think about, you know, endotherms that are all kind of sitting in around some pretty consistent temperature, so 35, 36, 37, you know, dogs I think are at 39 degrees Celsius as their average temperature. We have body temperature up here, and then we have environmental temperature ranging from -70, which is the lowest temperature in the Arctic, to, you know, just a little shy of 30, which is typical tropical average conditions. And so what happens is, if you want to maintain this body temperature, when you start to drop below a particular air temperature, then so this is our basal metabolic rate here, and when you drop below the temperature that allows that species to maintain that basal metabolic rate, all of a sudden the metabolic rate to maintain this body temperature increases. And you can see that this happens at different temperatures for different species. So here's our most extreme example, a sloth, a tropical mammal. It pretty much needs temperatures of just right around 30 degrees to maintain its temperature at this basal metabolic rate. Temperatures cooler than 30, all of a sudden we have that basal metabolic rate, we have that metabolic rate increasing. Okay, and the same thing here. But we can move across, and if you get to something like a ground squirrel or a polar bear cub, they're able to their basal metabolic rate doesn't increase until much cooler temperatures. These are species that are adapted, have adaptations for cooler environments, and are as a consequence able to maintain that body temperature to much lower temperatures. And so you can see that for a polar bear cub, it's right in here around 0 that it really starts to need to increase its basal metabolic rate to maintain its internal temperature. So different species have different responses. And you'll also note that the slope of the increase in metabolic rate below that sort of lower critical temperature for each of these species is steeper than it is for these these cold adapted species. And so not only is the lower critical temperature lower for these cold adapted species, these Arctic species, but the slope at which it increases is also lower compared to tropical species. And so it just speaks to the ability of these Arctic species to maintain their body temperatures at much cooler temperatures, much cooler environmental temperatures. Okay, so for some of these species, though, they need, temperatures simply get too cold during the winter for them to be able to maintain those temperatures. The cost, that metabolic cost of generating that heat at the very cold temperatures that we experience in our high latitude winters would be too much for many mammals, particularly sort of small bodied mammals, to be able to survive these winters. So when we think about sort of small mammals, their surface area to volume is very high, and so they have a lot of surface area to lose heat across. If food resources are insufficient through the winter, then one thing that a lot of these taxa will do is go into torpor or adaptive hypothermia. So what this means is that they basically allow their body temperature to decrease. And so their body temperature decreases, and their metabolic rate also decreases. And so what this does is it reduces the energy demand, the energy supply required for maintaining body function. And so all kinds of things are downregulated, physiological processes. Behaviorally, they act very, very, everything sort of slows down. The heart rate, the physiology of the organism slows down to a lower level. A lot of times organisms will feed quite a lot before going into these periods of torpor, which provides the energy to sustain them through that time. If they don't go into a full deep hibernation, their behavior will be modified so that they only periodically come out of sort of aroused to do certain particular functions. But what happens is the metabolic rate decreases and the body temperature decreases, sometimes by as much as 20 degrees Celsius. And so without the need to maintain that body temperature at that high level, the metabolic rate decreases. And then usually this is arousal here occurs when temperatures increase to a certain level, and then you can see that they come out of this state of torpor or state of hibernation much more quickly than they went into it. A sort of gradual entry and then a very rapid recovery. Okay, so just a couple of definitions then here. Torpor is a state of lowered body temperature, slow breathing, I should say heart rate, not hear rate, and low metabolic activity. So everything slows down in the organism. Hibernation, in contrast, hibernation is a sustained and prolonged period of torpor. So this is another amazing way that animals, that endotherms, are able to deal with these long cold winters that we sustain here. Okay, so we've seen lots of adaptations for dealing with temperature. Now what about water? So this is just a little quick review of water potential. You will have learned about this previously. So water potential, this is a simplified version of a water potential equation, but we have the total water potential being the osmotic water potential plus the pressure water potential plus the matrix potential, okay? So total water potential, osmotic water potential is the energy associated with dissolved solutes. So the more dissolved solutes you have in water, salts, sugars, whatever it happens to be, the lower the free energy of the water. And so osmotic potential is always a negative value. Any solutes reduce the free energy of water. Pressure potential, or turgor, is energy associated with the exertion of pressure, either positive or negative. And this dictates whether it's a negative or a positive value. So a positive pressure increases the free energy of water, and so it will act to move water from that area to an area of lower water potential. Whereas negative pressure potential, or suction, has it acts in the opposite. It reduces the free energy of water. It holds it more tightly, and so that will be a negative value. So pressure potential can be either negative or positive. And then matrix potential is the energy associated with the attractive forces of surfaces of large molecules inside cells or on the surfaces of soil particles. So this is a negative value. That attraction of water molecules to the surfaces of other large molecules or to soil particles reduces the free energy of water, and so this is also a negative value. So you basically have total water potential equals a negative osmotic potential, either a negative or a positive pressure potential, and a negative matrix potential. And it's important to remember that water moves down a water potential gradient from high water potential to low water potential. And free water, it has a water potential of 0. So anything that's not free water has a negative water potential. So when we think about water potentials, they're always negative values, okay? And so you have pure free water that has a water potential of 0. You add solutes to that, and this will have a more negative water potential than the pure free water, and so water will move from this pure free water into the area that has the solutes, so down a water potential gradient. Same thing, you put a positive pressure on this side, this increases the free energy of, so that's true. If you put a positive pressure on pure free water, then you have a water potential that's above 0. That's kind of the one example of when that happens. And so then water will move from this area of positive pressure to this area over here. And likewise, if you have negative pressure, if you put a suction on the water, then you reduce the free energy of that water, and water will flow toward over here. Okay, so solutes, negative pressure or tension, and matrix potential all reduce the free energy of water, reducing the water potential, okay? And this is really important for understanding how organisms respond to a number of environments. Notably, water balance in plants is really an important place to think about water potentials. So one really key thing to think about is turgor pressure. Turgor pressure is the pressure that the plant cell, so if you remember, plant cells have a cell wall, a rigid cellulose cell wall. The way plants grow, the way cells expand, and the way plants grow, and the way plants avoid wilting is by using turgor pressure, putting pressure on the cell wall. So if there's sufficient water inside this cell, then there will be pressure put on the cell wall. This allows for cell expansion. This allows for the rigid structure of plants. This turgor pressure inside the cell is really critical to maintain, and so we have this gradient in water potential that's established by the plant cell having more solutes than outside of the cell. So this is osmotic potential. So the osmotic potential value inside the cell is greater than that outside the cell. So water moves inside the cell. This creates a situation where there's turgor pressure imposed within the cell, and so eventually a balance will be reached. When the cell reaches sufficient turgor, that will balance this difference between the greater solutes inside versus outside. That's a negative value, and the positive value of that turgor pressure that is imposed by water entering the cell and pushing the cell against the cell wall. So that's This is a really important thing. These water potential gradients and the balance between pressure and osmotic potential are important for thinking about how plants function. Water potential is also really critical for thinking about how water moves through plants. So, the atmosphere, and they don't even have the value for the atmosphere out here. Okay, so this is not ideal. We should put this on here. The atmosphere, the water potential of the atmosphere is about minus 100 megapascals. Okay, so it's a very large negative value. Soils, in contrast, are very often very close to zero in terms of their water potential. So here we have a soil water potential of minus 0.5. We have an atmospheric water potential of minus 100, thereabouts. And so we have this water potential gradient, and then we have plants growing in this soil medium. So plants are connected to the soil and to the atmosphere. And so we refer to this as a soil-plant-atmosphere continuum. Soil-plant-atmosphere continuum, okay? And the reason that soil-plant-atmosphere continuum is important is because your soils are at a fairly low water or high water potential. Your atmosphere is at a very low water potential. Your plant is somewhere in between that. And so that water potential gradient from the soil to the atmosphere through the plant is what drives water movement through the plant. It's pretty amazing. So you go from the soil to the root. Some solutes in the root mean the root's at maybe minus 0.7 megapascals. So water moves from the soil into the roots. The stems are under a little bit more, have a slightly lower water potential compared to the roots. And this in part is because basically what's happening is that water is being pulled up through the plant to the leaves and out into the atmosphere because of transpiration. So the stomates are open. We know that the atmosphere is a very negative water potential. The leaves are, depending on depending on the time of day, depending on the soil conditions, the leaves are at a much higher water potential compared to the atmosphere, of course. So water moves from the leaves out to the atmosphere very, very readily. This is why transpiration occurs. As that water is lost from the leaves, it creates a lower water potential compared to parts lower down the plant. And so this basically pulls water up, almost like sucking water up through a straw. So there's, because of the cohesive properties of water, those water molecules just get pulled up through this water potential gradient from the soil, through the plant, and out to the atmosphere. And this is how we actually, these properties of water and the soil plant atmosphere continuum are what allow trees to get so tall. So it's all of these processes, and it drives the limitation to how tall trees can get. So then at night, what happens is the stomates close. So here in the day, the stomates are open, exposed to that very low or very high or low water potential atmosphere. At night, the stomates are closed, and then the plant becomes imbalanced with the soil. So the soil's at minus 0.5 megapascals, the roots are at minus 0.5, the stem and the leaves, they're all equilibrated. So if you take a water potential measurement on a plant at night, it actually gives you a measure of the soil water potential, which is a really great thing to know. Because the stomates are closed, the plant refills, and the water potential of the tissue in any part of the plant is equivalent to the water potential of the soil. Okay, and so we can look at how water potential changes through the day and in response to soil drying. If we measure water potential in the plant, the soil, and the roots. And so what happens is during the day, so here we have on the x-axis, the black bars are night, the open areas are day, And so you can see that during the daytime periods, transpiration increases. This is water loss through the leaves. The stomates are open. Water is being lost to that very low water potential atmosphere through the leaves via transpiration. And so transpirative water flux that drops down to almost zero at night, peaks back up, etc., etc. But you can see as we go through these days that this transpirative water loss is decreasing in response to this drying down of soil. So you can imagine an experiment where you had well-watered plants on day one and you withheld water from there on out. The soil water potential would drop. This is this red line. And what we can see is that the water potential of the tissue drops in relation to the soil drying. Now at first, the plant is able to maintain that water potential gradient, so water is still pulled up through the leaf and transpiration is maintained. When a plant reaches a point of when the soil is too dry, the plant is no longer able to maintain that and has to start closing its stomates in response to drought stress. And so this is where we start to see transpirative water fluxes decreasing. Okay, so this is how water potential works in plants. And plants have ways to respond. So first of all, of course, plants can close their stomates, and this is what is happening here. When the soils are too dry, plants can close their stomates and conserve water. Plants can also do all sorts of things like dropping leaves to conserve water. But when we think about biomass allocation, this is another thing that happens within species and across species, is when plants are exposed to drier soil conditions where water is limiting, they will allocate more of their biomass to root production, simply to allow them to access more water. They will do the same in response to nutrient availability. When they're shaded, they will tend to allocate more resources to above ground biomass to try to capture more light. And so plants respond in their biomass allocation to resource availability, and water is one of those examples. And so here we have a plot of average annual precipitation and the root to shoot ratio. So the root to shoot ratio is if you harvested a plant, measured the dry mass of its root, measured the dry mass of its shoots, so its stems and its leaves, and did the ratio of those. So root to shoot, a higher root to shoot ratio means more root biomass compared to shoot biomass, so greater below ground biomass. And so you can see where average annual precipitation is lower, we tend to have higher root to shoot ratios, more allocation of biomass to roots compared to leaves. As you increase precipitation, all of a sudden the root to shoot biomass becomes very low. There's a lot more above ground productivity, and this is typically because when those soil resources are not limiting, that means that plants are competing for light resources and they allocate more to above ground biomass. Okay, so biomass allocation tracks water availability. And this can be a plastic response or an adaptive response. Right? So if your plants in your garden are, not doing well in terms of water availability, they will not produce as much above ground biomass. They will tend to invest more of their carbon below ground. So that's a plastic response, that acclimatory response to reduce the stress. But we also see the situation where plants growing in these very wet environments have a very different root to shoot allocation compared to plants growing in very, very dry environments. And that will be an evolutionary response, because these are not gradients that you'd see in your garden. These are gradients at the global scale. So when we look at different plant communities growing in different parts of the world, those plant communities have genetically based differences in their biomass allocation to better suit those environmental conditions. If you grew them under wetter conditions, they would respond a little bit, but not very much. A Mediterranean shrubby plant is never going to, no matter how much you water, it's not going to become a big tropical tree. There are constraints there. So both plastic and acclimatory and adaptive responses to water availability. So there are some additional mechanisms of water exchange in animals. And so this is just a diagram that shows an aquatic larval stage and a terrestrial larval stage for a dragonfly. But there are similar processes that for these organisms that happen in many terrestrial organisms. So, we can think about losses of water and gains of water. So a key way that all animals gain water is through food and drink. So consuming water, consuming food. There's also both, uptake and losses across outer covering. In this case, this is the the exoskeleton of the insect. Where there are tracheids in that exoskeleton, and there can be water lost and water gained across that outer covering. Oxidative metabolism, so the breakdown of sugars using oxygen with the byproduct of water, and so this happens during cellular respiration. So oxidative metabolism, which occurs in all animals through the Krebs cycle. And then at this aquatic larval stage, of course, there's uptake across the gills and diffusion or secretion across the gills. So there's losses and gains across the gills. And you can think about many animals as well. When we're, for example, in humans, when we're breathing, we lose water to evaporation from those moist surfaces. So you can think about any example terrestrial organism and think about places where they gain water and lose water. But a key piece is gains through intake and a variety of losses across surfaces, and then this metabolic gain of water as a byproduct of breaking down sugars. Okay, terrestrial adult stage, similar kinds of processes. Similar kinds of processes as at the larval stage, with a few changes. So just acknowledging that for some organisms, as you move from a larval stage to an adult stage, in the case of insects, or from across different stages of development, that the gains and losses of water may vary. Hyperosmotic is where the aquatic environment is more saline than an organism's cells or blood. And so you've got more solutes in the water than in the organism. And so you've got an area of low water potential outside the organism and an area of high water potential inside the organism. That means the organism has the potential to the water is going to tend to move from inside the organism to outside the organism. And when the organism takes up water, it's going to have to do something with all the salts that are coming into the organism. We see this same problem in plants. This is just a really cool example of the fact that we do have plants growing in these environments as well. And so when they take up water, it's not fresh water from the soils, it's salt water. So this is a mangrove species, and those plants have to invest into specialized structures to actually secrete that salt so they can use the water. And so challenges associated with these hyperosmotic environments. Hypoosmotic is the aquatic environment is less saline than an organism's cells or blood, and so the water has a higher water potential, more free energy than the organism, and so water is going to tend to move from outside to inside, and so there's the problem of too much water. So plants face this problem as well, as I just exemplified, but we're going to talk about it here in some marine and freshwater teleosts, or fishes, so vertebrate taxa that live in these aquatic environments. And so when we think about marine fish, they lose water across their gills and in their urine. So water, again, water's moving from inside the body where the water potential is higher to outside the body where the water potential is more negative. So they're losing water across their gills and in their urine. And then they're drinking seawater that to replace this water that's lost, but they're taking in not fresh water but salt water. And so then they have a bit of a solute problem because they're taking salts up from seawater. So they have to be able to live in this environment, they actually have to be able to actively get rid of those salts. Just like we saw with the mangrove plant in the previous slide, these marine fishes have to be able to get rid of those solutes And so they have to spend energy to excrete excess solutes across their gills and in their urine. Okay, so this is a costly endeavor to maintain your to regulate your solute concentrations in your body when you're living in a saline environment. And likewise for freshwater fishes, they gain water across their gills and excrete excess water in their urine. So they're taking up water across their gills. They're excreting water from their urine. So water is moving from this area of lower water potential to inside their body to an area of higher water potential, in contrast to the fish in the ocean. And so they're excreting excess water in their urine. But they have this sort of opposite problem where there's not enough solutes because there's so much fresh water going in, and their solute concentration in their body is higher than that outside of their body, that they need to actively be taking up. So solutes are lost across the gills, and so they have to actively take up solutes in their food and active uptake through their gills, again, to be able to maintain their balance in their body. And so there are these challenges with aquatic environments of, that are associated with water potential gradients that drive challenges for these organisms. Okay, and as we already talked about, animals lose water in terrestrial environments too. A couple of points. We have a balance between gas exchange and water loss. And so we'll see in the next slide that if you increase the resistance to water loss in your body covering, so you can think about our skin and water loss through our skin, if you increase the resistance to that, it improves your situation from a water perspective, but it comes at the cost of gas exchange. And this is the same as the case for plants when they increase kind of waxy coverings in different ways to decrease increase the boundary layer around the stomates. This also acts to decrease the diffusion of gases into the tissue. It reduces the loss of water from the tissues, but it also acts to decrease the diffusion of gases into the tissues. And so same problem for terrestrial animals. And then high water loss can be balanced with high water intake. So, when you're hot and thirsty, you can always drink more water, but that's great until your water supply fails. And so, these constraints limit where different organisms can occur and the types of environments they can live in. And different animal groups have different abilities to tolerate water loss. And so you can see that the range of water loss that different species, here we have invertebrates, that they can tolerate is really wildly variable. So frogs can tolerate 28 to 48% weight loss in terms of water loss, whereas humans can only tolerate 10 to 12%. And this, these things will vary within any of these groups. So some rodents we'll see a rodent that is able to tolerate a lot of that has a very high tolerance of, that has really creative adaptations for minimizing water loss and being highly conservative and efficient in its water use to avoid water loss. And so we have some species that are just able to deal with a lot of water loss, and other species that have to develop adaptations to be able to cope with that stressful environment. Okay, so as I mentioned, this resistivity of external coverings is key to reducing water loss and can vary by environment and by taxa. So here we have a situation where if we expose different species to desiccation, we see very, very different responses in terms of weight loss. So this is weight loss of water weight loss. And so you can see these toads and frogs, so Bufo, Rana, Xenopus, these all show these steep decreases in weight in response to exposure to dry conditions. Their skin resistance is low. They have very permeable skin that water evaporates very readily from, and so they have very rapid water loss from their surfaces, so the resistance to water loss is low. In contrast, this Chirotomus frog has a higher skin resistance. Similarly, this Cameleo lizard has a very high skin resistance, and those species were able to maintain their body mass despite ongoing kind of drying conditions. And so you can look at this this resistance across different groups, and so you see some patterns where there's, you know, fairly low resistance across many taxa that occur in wetter environments, so fish, frogs, earthworms, and then we look at things like desert lizards, desert scorpions, and their resistance across to water loss across their skin is very, very high. And this, of course, comes at a cost of gas exchange. They have to develop other adaptations to be able to cope with that. But when we're talking about desert conditions, it's really important for those taxa to minimize water loss in these incredibly dry environments. Okay, and these kangaroo rats are an extreme example of evolution of drought tolerance. So they're just able to cope with dry conditions. They use we have their water balance here. So gains, most of their water comes from oxidative metabolism. So breaking down sugars, converting them to energy, and the water that is produced as a byproduct of that. And a little bit comes in their food, but they're living in these extreme dry environments, and so very little water in their food resources, and so they consume a lot of fatty seeds and things like that, and use oxidative metabolism for most of their water intake. They also have very thick, oily skin with very few sweat glands, and so they really minimize their water loss across their skin. They use behavioral changes, so they move from they spend most of their time foraging at night when it's cooler and at peak humidity, and spend their days underground in their dens where it's where they're shaded and where it's cooler and more humid compared to being above ground. So they're nocturnal organisms in terms of their activity, and they have their quiet time during the day. And then exceptionally efficient water removal in kidneys and intestines. In fact, this species has the most concentrated urine of any species on the planet. And so urine concentration is a really great way to minimize water loss. If you have really effective uptake of water or reabsorption of water in your kidneys and your intestines, then you're able to minimize your water losses. And so you can see their water losses to urine are very, very low compared to what it would be for many organisms. Okay, and then back to our original picture from the start, from the first slide, which was a picture of a frog that was frozen solid. And I like to think about these trees, these boreal trees, growing in minus 50-degree temperatures in some cases. They face similar kinds of stresses, and so there's unique challenges of freezing that go hand in hand with temperature and water. So there's sort of water-related challenges that these species that are able to deal with freezing have to manage. And so some of the major challenges that are that go hand in hand with this particular problem are the formation of ice crystals inside the cell. If you form ice crystals inside a cell, whether it's a plant or an animal cell, that cell will typically be killed because those ice crystals puncture the cell wall, or the cell membrane, and the cell dies. So there's all kinds of neat adaptations that these species, whether plant or animal, use to avoid that. So in both cases, extracellular freezing of water, so making sure that the freezing water, if the water is not freezing inside the cell, but it's freezing outside the cell in the intracellular spaces. And ice nucleating proteins that are formed outside of the cell, again, to control the formation of ice, so that ice formation happens outside the cell, not inside the cell. Within the cell, these taxa synthesize different antifreeze compounds like glycerol to lower the freezing point within the cells. This also helps to stabilize the cell volume, so the cell doesn't just kind of collapse because all of the water has moved out of the cell into the intracellular spaces. The production of these antifreeze compounds increases the volume of compounds in the cell. And in terms of restriction of oxygen supply, because of course, you know, as you shut down, you have your metabolic processes have to change, because you don't have the same kind of, you don't have the sort of diffusion that is happening typically when you're not frozen. So in all these cases, there's physiological changes that act to slow metabolism, and then upregulation of genes that help to protect the cells from damage during these stressful times. Okay. That is all I have to say about temperature and water today. We will continue this conversation about the response of organisms to environmental stress in our next lecture this week, and I look forward to seeing you at student hours tomorrow. Have a great evening. Bye.